will be very robust in terms of analysis of single cell data. And again, this is mirroring exactly these ideas of trying to find the axes of cellular identity. So we hypothesize this linear sparse optimization is going to give us a way to find these axes of cellular identity to relate back to therapeutic response. Now, I've sort of given you all this theory into that, but the trick is, how do I know if any of that is real, right? And you know, it's very easy, and I think we do this a lot as a computational biology community. It's very easy to come up with computational metrics to evaluate when our systems are performing well, right? We can look at the L2 norm or some other norm, see have we minimized some cost function and go, aha, our method performed great. But then we need to really go back and take a step and say, okay, what is the biological validation? How do we develop biologically driven metrics in order to interpret if these systems work well? And there, the big challenge in biology, of course, is that we lack a ground truth, right? So this would be very easy if we knew the fundamental truth of biological systems. But again, if I go back to sort of my vision of all this and where I'm coming from, you know, in terms of thinking my background, those biological rules are really the big black box. We really don't know what those are. But we're fortunate in that there are some systems, you know, cancer biology is not one of them, but there are systems, for example, in developmental biology, where the characterization of how the cell types are sort of developed has been very, very well described for a long time. And the other advantage in developmental biology is that, unlike cancer biology, where the whole system gets completely rewired, developmental biology is very constrained, or else the organism wouldn't survive and the phenotype wouldn't develop. So as a benchmark to our methods, what we did is we first took single cell data of the developing mouse retina. And the advantage of the mouse retina is that it serves as a more complex, it serves as a simplified model for the whole central nervous system development because it contains a lot of similar cell types. And again, the order and timing of the developmental processes of the cells, as well as many of their key drivers, are already known. So my colleagues, Loyal Goff and Seth Blackshaw, led by a new assistant professor, Brian Clark, went out and they went for the developing mouse retina and performed single cell sequencing of over 100,000 cells spanning different age points and different cell types in the mouse retina. And this here is a two-dimensional UMAP of the data, or three-dimensional, sorry, UMAP of the data. And so if you look at the sort of duck representation on the UMAP of these data, what you can see is that on the left, if you know a priori the feature you're interested in is age, you can see that the UMAP does a very good job of segregating age. It also does a very good job on the right of segregating cell type, if you know cell type. But going in and making these colors and groupings and clusters relies on a priori knowledge of the system. And it doesn't get at sort of the cell state transitions that might be most interesting in terms of looking at the developmental processes that we really want to validate. So in order to do that, we took this single cell data and we applied our COGAPS method to it. And when we did that and applied the factorization, we found 80 total patterns that were associated with the data. One of them, and several of them, we noticed were enriched along very specific regions of the UMAP that could then be associated with cell type. So for example, this here is a pattern that was very enriched for rods and comes up in rods. There were others that were more diffuse across the UMAP and across different stages that were associated with temporal progression. So for example, this was a cell cycle pattern on the right that was associated with the sort of differentiation of progenitor cells into their final phase. We also observed we observed patterns that were associated again with different cell types. So we separated rods We separated out precursor cells. We separated out cones, all the sort of different cell types you would want to see in different patterns. And we also separated out temporal patterns, again, that were associated with neurodifferentiation, neurogenic transitions, and cell cycle changes associated with these with the progenitors. So within this one factorization, it gave the advantage that if you looked at the system across different dimensions, you were able to see different features that were either associated with cell type, cell state transition, and others that were associated with technical artifacts. So the next question that I always get asked is, how do you know that any of this is real, right? Like, how do you know any of this is meaningful whatsoever? You know, you see these nice, blobs that are lighting up, but how do you know what's going on? So in order to do that, again, sort of thinking about how do we get this biological benchmarking and validation? What we did is we thought, okay, if the patterns that we're learning are real, then the genes that are associated with them that we learn should occur in other data sets of similar contexts. Whereas if they're purely technical, the patterns that we observe won't work that way. And they'll really they shouldn't project into the new data set because it won't have those same features. And so what we imagine is that these patterns that we've learned through the matrix factorization, we can think of them like they are, like they're lenses through which we view the data. And then you can take a projection where you take a new data set, shine it through the lenses by saying, okay, if I have the gene weights, how do these project onto a new data set? And see where they occur in a new data set. So this gives you the ability to do validation. It also gives you an ability to query how much does a feature occur in a new target data set. So to validate this data, our data set was the developing mouse retina. There was a mature mouse retina data set by Moscato in the public domain. And so what we did is projected our data set onto this adult retina, with the idea being that mature cell types should preserve, whereas the patterns that are associated with the early developmental processes won't. And that was exactly what we observed within this data set. So I'm showing you one example on the bottom, this pattern 21, which was associated with rods, projected perfectly into the adult murine data set. This projection was observed even though the data was from a different strain of mouse, different single cell measurement technology. It was basically everything you could say in batch effects you don't want to occur, but it still worked. Whereas the patterns that were really associated with the early developmental and transitory processes in development did not project onto this adult data set. Neither did those that were technical. The other thing that's exciting about this is because this projection technique just uses gene signatures, you're not limited to looking within one organism. You can also go across species. And so we applied the same approach to look at the development of the retina in the mouse, or from the mouse, to see how it relates to the types of development we see in the human. And we observe these patterns that are associated with cell state transitions also occur in the same way in the human data. And some of the terminal cell types are enriched in the same way within the human data. So this gives us an ability to look at what's preserved across species, which for cancer biology has the exciting capacity for me to think about, okay, if I'm learning these therapeutic responses, what are the things that I'm going to be able to go back and validate in the model system? And what are the things that I'm not, right? So what's really going to be unique to the human response to therapy? And what are the things that are really going to be robust? The other thing that was exciting about this was that we weren't limited to applying this approach to single cell RNA-seq. We could also apply what we learned in single cell to bulk RNA-seq time course data. and start to deconvolve the ways these different processes are occurring within the bulk data. So once we benchmarked, then I'm going to go back into my picture of cancer biology. So how do we sort of use this in a therapeutic context, right? Now that I know it's working and I'm finding these cell types and cell state transitions, what can I do to sort of evaluate how this works in cancer? So in order to do that, a grad student of mine, Emily Davis-Marsasak, went through and collected public domain single cell profiling of immune cells from a preclinical sarcoma model that were treated either in control or with two different immunotherapies, anti-PD-1 and anti-CTLA-4, either alone or in combination. And the standard clustering analysis that had been done in the original manuscript of these data found that they sort of found the typical cell type clustering and differences in cell types that were associated with therapy. When Emily applied COGAPs to these data, she found additional sources of heterogeneity. So some of the patterns were really associated with cell type and cell lineage, but some of them had more of a gradient. For example, this pattern 7, which it turned out was activated in a subset of the NK cells and was associated with NK cell activation. So again, our COGAPs approach is delineating heterogeneity in NK cells. When she went to the mouse data to look at it, what was exciting is that this pattern of NK cell activation uniquely occurred within the cells that were treated with anti-CTLA-4. And so that led us to hypothesize that activation of NK cells was associated with anti-CTLA-4 response. To further validate that this was relevant, right, because this had been shown in mice before, she went through and took this same pattern and applied our transfer learning approach to relate it to the response that was observed in humans, in human melanoma patients treated with anti-CTLA-4. And she found indeed that NK cell activation associated with differences between responders and non-responders who were treated with anti-CTLA-4. That led us to go back and do further experimental validation with molecular biology. And Emily and Allison showed for the first time that CTLA-4 was activated on NK cells, on the surface of NK cells, and expressed in human cells that are associated with response. So as I sort of get through, that does a really good job. This sort of single cell technologies do a really good job of getting at cellular composition and their contribution to therapy. If I go back to my original cartoon, one of the other things is it's not just what cells are there, but it's what is their spatial distribution that's going to be really critical to therapeutic response and resistance. For example, if I have a ton of activated T cells within the tumor, I could have the same composition in B with all the T cells inside that would have a totally different phenotype than with them spatially surrounded. And I think we're fortunate today that spatially resolved single cell technologies are really the forefront of what's coming. They're really the forefront of new technologies. And we have, at the same time, proteomics single cell technologies, largely imaging based, but some sort of mass cytometry based. These have the advantage of very high cellular resolution, but relatively low molecular resolution. But you get very robust staining for specific antibodies of cell types and functions, so you can really query what you're looking for. Spatial transcriptomics technologies are also emerging. There you get a trade-off between molecular and cellular resolution, but we're coming to a point with technologies such as Visium from 10x Genomics, where we're advancing to whole transcriptome cell-level profiling, which is very exciting to think about these relationships. So as preliminary data, we formed a collaboration with 10x Genomics to start looking at Visium data in invasive ductal carcinoma tumor. And so traditionally, what we would have gotten from imaging data is something like on the left is an H&E slide, where we can sort of delineate different regions that have immune cells or that have invasive lesions. And that's what's shown on the left. In addition to just the H&E, The Visium technology also gives gene expression at a not quite a single cell level, at these little spots that are close to cellular resolution. Maybe you get up to 30 cells, but you get whole transcriptome profiles. And so you can start to overlay and see, for example, on the right, see specific populations of immune cells. So on the right, I've drawn CD8 as one marker of T cells. So the question is then, how do you interpret these data where, again, you have this spatial and molecular information? So no surprise, I'm going to go to our favorite COGAPS approach and apply this to look at these data. And when we do this, what was exciting was that we found different patterns completely unsupervised that were associated with the different lesions and invasive carcinoma in the tumor. The other thing that was very exciting to us, and a little bit terrifying from a clinical standpoint, was that each one of these lesions, if we looked at it, had a different dominant molecular driver and molecular signature occurring. So for example, the invasive carcinoma is largely driven by TGF-beta, VEGF, and FGFR pathways, whereas the other invasive carcinomas, the sort of smaller lesions, are associated with other growth factor receptor pathways. So for example, we observe, or hormone receptor, we observe insulin receptor as a dominant signature of this lesion, androgen response as dominant in this lesion, and estrogen response with this lesion. So this suggests that the sort of underlying heterogeneity that we've been seeing in tumors and the rapid outgrowth of therapeutic resistance is fundamentally built into the transcriptional signatures that occur within the different lesions within a tumor. So to sort of summarize, matrix factorization techniques are incredibly robust tools for biological pattern discovery from bulk and single cell data. Our COGAPS method is a unique package in being able to recover biological meaningful patterns. This idea of transfer learning with our software ProjectR enables us to explore biological process reuse and how much they're occurring between experiments, different measurement technologies, tissues, species. We've recently shown that it can relate RNA and ATAC-seq, and also that the transfer learning can be used to integrate single-cell RNA-seq with imaging data. And finally, this pattern detection technique is really robust at delineating therapeutic response in genomics and imaging data. So with that, this is only sort of a subset of my collaborators and members of my lab who really contributed to the work that I presented today. So Genevieve Stein-O'Brien really drove a lot of the analysis for the retina data and developed the transfer learning methods. Tom Sherman is responsible for COGAPS being computationally efficient enough for single cell data. Emily Davis-Marsasak and Gaurav Sharma really worked together on a lot of the applications to immunotherapy, and Melanie Moff has been working on the Visium data. So with that, I'd like to thank my collaborators and funders, and I'm happy to take any questions.
Thank you very much, Elaine, for this nice and interesting talk. And as Elaine said already, that she will be also interested talking to participants about the career and not only the scientific or pure research, but other career-related questions also. So I'll start with the question that you described the ProjectR. So in ProjectR, you are doing UMAP once, and then you're projecting the UMAP values in the another dataset UMAP value?
We're not projecting UMAP, we're projecting the features. So I'll go back to the slide. So we have the matrix factorization that we're using. So we have our COGAPS approach. And so the idea is that you apply COGAPS in order to figure out the different gene signatures. So however many dimensions from the non-negative matrix factorization. And then what it is you project the gene weights that you get from the matrix factorization onto the new data set. So the only way we're really using the UMAP is to visualize the weights of the projected patterns. So for example, in this data set, right, we learned 80 different patterns. We would project each one of them, for example, the rod pattern in the case I show here, we would project that, but all we are using the UMAP for is to visualize the intensity of the projection for that particular pattern. We could go through and do it for any of the 80 that we learned, if that makes sense.
Another question is that a common problem in single cell data is having the dropouts, or sometimes we can't measure certain genes in certain samples. So how do you handle that, or do you have any opinion about that?
So I think it depends on the analysis technique that, that you are trying to apply. So we found the matrix factorization approach, because it's explicitly modeling sparsity, to be very robust to the dropout, and that we don't really need to do much for it. On the other hand, we've also developed another technique called EVA, where we quantify sort of the transcriptional heterogeneity of profiles. And we found that was incredibly sensitive to the missing data, and in that case, you needed to impute. So my feeling is that it really depends on what analysis technique you want, and what sort of the underlying model is of that technique, whether you want to impute or whether you want to work with the raw data.
So if you look at the Zoom Q&A, you can see there are already a few questions. So I would read the second question first. How is the label transfer method in ProjectR different from that of Seurat in terms of performance?
Yeah, so we haven't done a head-to-head performance with Seurat in terms of that. I think that both of them, so there's sort of three dominant techniques that come out, right? There's coupled matrix factorization, there's Seurat's canonical correlation analysis, and then there's the transfer learning approaches with ProjectR. In terms of the coupled matrix factorization technique, one of the things that can be limited is you're really enforcing that specific genes or specific cells are lining up in the same way. And so, for example, if you have time delays, that might not work well. Seurat and ProjectR both handle that well because they're sort of transferring to a different system. I think that Seurat, you still sort of have this assumption that the anchors are going to be shared in both. And so we think that ProjectR might be most robust in the case where you have some processes that are preserved, but some aren't, and you want to sort of find what's unique and what's different to them. But we haven't done a head-to-head comparison.
Okay, so another related question, the first question that I would be interested to hear more about comment you made that a pitfall computational biology research often falls into using actual biological data to validate new method. Sorry, not using actual biological data to validate method. So can you comment on that?
Yeah, I mean, I think that is a big limitation in the field. You know, I think a lot of times we, you know, focus on some sort of a cross-validation metric within the same data set, or, you know, some sort of a computational metric. I think we're fortunate these days that there's so much public domain data that we have the ability to cross-validate across public data from, you know, related systems and see how well they hold. I think there's sort of the converse problem in the biological community that I don't think they trust any sort of in silico validation. And so in the end, there's like still this need to go back and do an experiment. I think, you know, as a community, both on the biology side and the computational side, we need to come together and sort of recognize, you know, the value of cross-study validation for benchmarking and really believe in it more than, you know, a new experiment that's much lower dimensional. But, you know, I think it's early days, and, you know, it's really important to think about what are the benchmarks that we're doing, and, you know, what are the benchmark data sets? What do we need? And so we just had a multi-omics workshop, and one of the big discussion points was, what is it that we actually need to validate as a community, to get ground truth? What is the data set we would like to see? And I think that that's something that's still very much open.
Okay, then there is another question that when you are you incorporating any homology metrics when you're projecting the gene weight across species?
We have not, and we've really just done sort of the stupidest thing of taking the genes that are most, similar between the species and projecting to that subset. So I think the whole area of cross-species analysis and how you map genes is an incredibly, ripe area of research.
Mm-hmm. Okay, so another burning question that I have is about validating these methods or models because you're working with the immunotherapy, and it's always difficult to get the clinical trial or something like this to translate these methodologies into the clinic. So do you have any opinion about that?
Yeah, I mean, I think that's certainly a big challenge, right? We have a lot of public tumor data, but very little public tumor data with actual treatments. And I think that's something that, for myself, I've been able to get around. I have a lot of good collaborations in terms of starting to get those data. But it's a real challenge for the larger comp bio community, where, because those data sets are, developed by pharmaceutical companies or by, clinicians whose bread and butter is these trials, it can be very hard. There's not sort of the same culture or ability for data sharing at all times because of sort of the IP, intellectual property, and, and potential for, financial gain from some of these analysis methods. And so I think one of the things, again, as a computational cancer biologist, that I would love to see advance and is starting to, is more data sets from these trials that are really made available to the computational community for analysis in a very real way to start exploring this. Because I think data availability is a huge issue.
Thank you. And then we have another question here that if we find different patterns in the invasive cancer cell in terms of different receptors responsible for tumor growth, how do we choose an appropriate therapy?
Yeah, I think that's the $1 million question. So, I think we would all love to know that. I think, one of the big ways, I think that's where the experimental collaboration comes in. So there's a lot of work with organoids and other systems of maybe, just because I showed all those different pathways are there, don't mean they're growing out the fastest, right? And I think that's where we have a lot of work to do from the experimental side of maybe you could take each of those subclones, make an organoid, see how they grow experimentally. This may also be a role for, sort of our colleagues doing more mathematical biology and mathematical modeling. If you have these and you have information about cell growth coming out from single cell data, could you also model how quickly each of those are growing and select therapies that are dynamically changing with the patient's response? So that would be my bias. But I think we don't know, and I think, because if you look at that, right, I think the answer that most clinicians would give today is throw a huge cocktail of drugs onto that patient, but that's going to be highly invasive and have huge side effects.
Okay, thank you. So we are very near to the time, and I will ask the last question, which is not research related. So as we are focused more on the students, PhD postdocs, and students in the computational biology. So what would you like to share your secret for being successful in computational biology?
Wow. So I think, when I entered the field and where I entered the field too, computational biology was viewed, I think, more as a service to interpret data rather than its own independent science. And for years, people always ask me, why are you doing matrix factorization, right? You can run a differential expression analysis in bulk data and get out all you want. I'm like, yeah, but we're getting these really interesting patterns that are coming out. And so I think some of the advice that I would give to people is, learn what the standards are in your field, learn how to do them, and learn how to be flexible to kind of adapt your computational methods in a way that really gears around the biology, but also stick to your guns in believing in your methods. And just because your methods seem kind of out of the box to start with, that might be a good place to be, and if you have that solid grounding in biology, you can work as part of collaboration to take these methods and start making them more and more biologically interpretable, to clinicians. And so I think having a foot in both worlds where you know the biology well, but you stick to your guns in terms of, some of the theoretical and computational advances and keep going is really important. And it's hard to balance both. I mean, finding the time, and personally, I always find it easier to balance, projects for other people than for myself. But, it really is critical, I think, to balance both sides.
Thank you very much, and thanks for the nice presentation and your advice. And I would like to thank you on behalf of SCS and ISMB 2020. So